Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host for today's webinar. Today, Dr. Daniel Geschwind will present Using Genetic Maps of the Brain to Understand Autism and Related Psychiatric Illnesses. The Brain and Behavior Research Foundation funds the most innovative ideas in neuroscience and psychiatry to better understand the causes and develop new ways to treat brain and behavior disorders. These disorders include addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, obsessive-compulsive disorder, post-traumatic stress, and schizophrenia. Since 1987, the Foundation has awarded more than $418 million to fund more than 6,000 grants around the world. I'm delighted to introduce Dr. Daniel Geschwind. Dr. Geschwind is Professor of Neurology, Psychiatry, and Human Genetics, Senior Associate Dean and Associate Vice Chancellor of Precision Medicine at the UCLA School of Medicine. He was a 2015 Distinguished Investigator Grantee, a 1999 Young Investigator Grantee, and the 2012 Ruane Prize winner for Outstanding Achievement in Child and Adolescent Psychiatric Research. Today's webinar will begin with Dr. Geschwind's presentation. This will be followed by a question and answer period. To submit your questions, please use the Questions tab on the control panel. Please feel free to submit your questions at any time. Following the presentation, I'll ask as many as possible in the time allotted. Now I'm pleased to introduce Dr. Geschwind. Daniel, the floor is yours.
Thank you so much. I just want to say before I get started into my talk how important the BBRF, former NARSAD, has been in my career, and I think in the field. I just want to, obviously, Jeff is, directing this thing, but take it from me as somebody who's outside. The foundation has played an outsized role in helping young investigators get started, which was the case with me, as well as pulling the field together and allowing us to do innovative work that otherwise we wouldn't be funded to do. So it's really with great pleasure that I'm here today to discuss some of my work with you, because I just have such a warm place in my heart and fond feeling for the foundation and what it's done for the field. So as I just want to, I have to, my usual conflicts of interest. None of the stuff I'm going to talk about today is really related to these, but I put them up anyway, just to be as transparent as possible to start with this. And I also like to start with my acknowledgement. So you can see my lab on the right. At some point, they're all, you can, a little tongue-in-cheek, everybody is taking my same pose. I've really had the reason I put this up first is just to say that the work and the kind of work that we do, and I think the most innovative work going on today, often involves a village. It involves multidisciplinary work involving multiple different types of expertise. And so the work that I've done relies on collaborations with statisticians and computational biologists like Steve Horvath, Bogdan Pasanuik, Sham Prabhakar, other collaborators at Stanford, Sergio Pasca, Thomas Weria and Andrew Schork in Denmark, running a huge population cohort. And then the work that's been done in my lab are the folks in blue, many of whom have gone on to now running their own laboratory or in training otherwise. And those people are starred. And I'll mention these people as we go forward. And again, the direct funding for the work that I'm going to talk about today is mostly NIMH, which has funded the Brain Initiative and Encode, and a little bit from the Paul Allen Foundation, as well as the Lundbeck Foundation. So what I want to share with you is that we're in the midst of a genetics revolution that's transforming our knowledge of disease. And this notion of precision health, I'm going to briefly introduce, because I think it's relevant not just to autism, but broadly to the field of mental health. I'll talk a little bit about what we've learned from autism genetics at this point, what we've learned about autism from genetics. And the kind of theme of my talk is a kind of, maybe difficult one for lay folks, and I'm going to try to make it simpler, but please ask questions. That most of the, only 4% of our genome codes for proteins. Those are the business end of the genome. The rest of it, the 95%, is all, you could call it a control center. It's an area that, 95%, which turns genes on and off, and kind of leads to higher order regulation. And so genes are exquisitely regulated, turned on and off, and that's where most genetic variation lies. And so understanding the mechanisms through which genetic risk factors act necessitates understanding which regions of the genome are active in which tissue at what time and what cells. And I call that a map. And it really is a map. And you can imagine that we're going into this area 5 years ago without any maps. So you can imagine how difficult that kind of navigation was. And now we've been building maps that have been transformative in allowing us to understand how genetic risk for disorders acts. And what I'm going to say, if you want to then go read the New York Times or something, is that these maps implicate early cortical development as a substantial period of risk for both autism, which is an early onset disease, maybe not surprising, but as well as later onset mental health disorders. And so, and I'll take you through that later in the talk. But our whole idea is to use genetics to understand the mechanisms of these disorders, what are the causal factors, and then understand how those causal factors act in the brain. But first, I'd like to introduce more broadly how genetics and how we're moving in the midst of a revolution in healthcare. Medicine today is reactive, population-based, one size fits all. And you come in for hypertension, your doctor prescribes the thing they're most used to. They don't tailor that to you. Precision health, the notion of it is to be predictive, so preventive, and a patient-centric model of care. What's the goal? And I think it was best stated by Obama: deliver the right treatment every time to the right patient. And of course, this is made difficult because prediction is very difficult, especially if it's about the future. I always love that quote from Niels Bohr, the Danish physicist. And again, so one theme will be, prediction isn't 100%. That's really part of it. It's probabilistic. And so what we're trying to do with genetics is understand if we can separate people at very high risk for something before they get it, prevent it, or using the genetics, find the cause of somebody's disorder that they already have, and use that to specify their treatment. And already that's happening in cancer, where about 6% of tumors can be sequenced, identify mutations for which targeted therapy is indicated. So the major goals are diagnosis, prediction, that is asking about your disease risk and potentially outcomes, prevention, of course, a big deal. And as part of that, the optimizing outcome, one has to optimize treatment choice, both to optimize your benefit and minimize any adverse effects. Another piece of this, especially for most disorders, especially for mental health disorders, is that until genetics really came along, we did not understand that well disease, what we call pathophysiology, or the causes. And that's a kind of major piece here as well that we're learning. And I'm going to talk about that quite a bit. Having genetic information can help determine best response to treatments. Most drugs prescribed in the US are effective in less than 60% of treated patients, and that's just a fact. And so we can do better than that. Now, what this means, though, it doesn't mean that immediately on having your genetic information, I'm going to be able to make a decision. There's a whole bunch of research we need to learn, and we combine big data from patients with genetics to move this forward. And I call that a learning healthcare system, where the blurry boundary between research and clinical care really exists. There are two major drivers of this, as I alluded to: dramatically decreasing DNA sequencing costs and computer power increase. So what we see here in green is the Moore's law. That is that Gordon Morse, the founder of Intel, stated every 18 months the price and density of an integrated circuit is going to double. That's an exponential change in computing power. That's the green line. That's exponential. This is a log plot on the left. Look at what happens with sequencing costs. There's an inflection point where it actually drops below $1,000 a whole genome. That's because of disruptive technology. And now a whole genome costs us somewhere in the $600 range to actually do. And we usually sequence the protein coding part of the genome called the exome because we can interpret that. The best and many mutations reside there, large effect mutations. So that's typically what's done in practice, and I'll describe that. And again, this explosion of computational power. Now, this change in sequencing could not have occurred without the change in computation. Computers have really, and miniaturization and other kinds of technology, it's really driven this. So it is a technological and very highly coupled technological revolution. Millions of genomes have been sequenced, and we're going to have the power to analyze them. That's critical. So cost of genome sequencing continues to drop rapidly. This is, it's continues to drop. Many more genomes sequenced. And what that means is we can now understand an individual in the context of the population. In other words, to know what your risks are, I can't just study you. I have to know, okay, you have this set of variants. Those sets of variants protect you from diabetes. These increase your risk for heart disease, etc. All of that is done by understanding populations. So the idea is new drug targets, prevention, diagnosis, and prognosis, optimizing treatment. So what is the nature of the genetic factors that are contributing to this? And so I'm going to give a very brief kind of primer on this. I'm going to dichotomize genetic effects in our genome. So we have 3 billion base pairs of DNA, and I'm going to dichotomize the kinds of mutations that can occur into two things. One is mutations, which have very large effect size. And what effect size means is that that is likely to be causal, that the effect is large enough of having that variant so that you can call that causal. Without that, you wouldn't have the disorder. So this would be rare recessive disorders, things like Down syndrome, and as I'm going to show you, about 20% of autism. But those things are very low frequency, as you can see here. The high frequency things, the things that you and I all share, that's present in more than 1% of the population, that defines common. Those have small effects, but they contribute to disease in an additive fashion, kind of in aggregate. So no single variant there has a large enough effect for me to predict that you're going to have the disease, because it doesn't cause disease. It just incrementally adds to your risk and et cetera, or protection. So if we want to think about that common variant space for a moment, think about it as one conceptualization is a normal curve that the average person has an average risk. And as you carry more and more risk alleles, a risk allele would be a copy that is increasing risk. You have more and more risk. And if we can identify the 5 or, this top piece here, either the top 5% of risk or 1%, that effect size can be 5 to 10 times risk of somebody who's in the middle. So it can have the aggregate effect of hundreds or thousands of common variants together can give you as much risk as one single rare mutation. And it's likely, I've dichotomized these things, but that likely in many cases, it'll become kind of a combination of both the common genetic variation, the kind of things that, are related to our height, our weight, our blood pressure, all of that kind of stuff also contributes in the same way to our mental capacities and behavior. So, and just to ground you a little bit, that there is a striking genetic contribution to major psychiatric disorders. So number one on the left, these prevalence of these disorders is high. Autism is in green towards the bottom, around 1%, close to schizophrenia, but you can see that many of these ADHD, anxiety, major depression are extraordinarily common. If we do family studies and twin studies to ask about how heritable are these conditions, they're all highly heritable. There's a range. There are both environmental and genetic contributions. Don't get me wrong, I'm not a genetic exceptionalist. It's just that genetics is concrete and predictable, and I can measure it. Now, if I measure the common variants that people have, so-called single nucleotide polymorphisms, or SNPs, these are genetic variants that aren't causal, that are present in more than 1% of people, I can kind of get an estimate for how much of the contribution contributes to disease of heritability. And again, that tells you that quite a bit of these disorders, including for something like autism, shown in green there, is substantial. There's a lot of heritability that comes from common genetic variation. But most of the advances that have occurred to date have occurred in the rare space. And there's a reason for that. It's because it's a little bit simpler. And another thing that we've learned from these data is that if we use the SNP-based heritability, if I genotype people with autism or schizophrenia or bipolar, and then ask, what's the correlation of their genetic markers outside of any familial, we take away any familial resemblance, that kind of, and now I ask, what's the correlation? We see that these disorders, even autism and bipolar and autism and schizophrenia, share non-zero amount of genetic risk. In other words, there's both shared and distinct risk for each of these disorders. And so part of the complexity of what we have to do as scientists, once we identify genetic risk factors, is understand how they're acting, understand how they produce risk for different disorders in many cases. How does this translate to clinical practice? Well, right now, we're not really using the kind of additive genetic risk, because as you could see, it's kind of on a normal distribution and, it's in the gray zone. What we do is we do genome and exome sequencing to find these rare causal mutations. And so this is just a paper that we published, my colleagues at UCLA this was a group that was really one of the first in the nation about almost a decade ago now to start doing exome sequencing in rare disorders. So about 7 or 8% of the population have disorders that are present in less than 1 in 1,000 people, rare disorders. So even though rare disorders are individually rare, they compose together a substantial amount of illness. And if we look here, you can see the rate of diagnosis without any other test, the single test of exome sequencing is over 25%. It's the most high yield clinical test actually in action, when applied properly. And these rare, what we call Mendelian forms of disease, are estimated to account for about 7% of patients. So a lot of what's happened over the last decade, very, very rapidly, is we use genome sequencing to identify people with rare forms of disorders. And of course, this has significant implications for treatment and recurrence risk. Is this a new mutation? Is it like Down syndrome, where the parents don't have it, it's in the child? That's called de novo. But knowing how it's inherited can be really important in terms of counseling and knowing whether there actually is risk. If you have one child with Down syndrome, it doesn't mean you're at increased risk for another one beyond the regular population risk of age and other things like that. But if it's an inherited mutation, that's a different story. And of course, we can identify mechanisms of disease and target therapies. You know, we're discovering several dozen new diseases at UCLA a year now. And some of these we're able to actually come up with a treatment based on identifying the gene. There's an obvious drug out there that already targets it, et cetera. Or sometimes we have to start, you know, that's where the research gets started, right? Starting to understand what the mechanism of that, how that gene acts. And the other issue is early diagnosis and intervention to alter trajectories. So now I'm going to kind of put this basic primer to act within autism, okay? So autism is not like other mental health disorders or neurologic or brain disorders. It's a heterogeneous neurodevelopmental syndrome. That is, it's not defined by its etiology like tuberculosis or specific forms of bacterial pneumonia or COVID. And so it's a syndrome. It's diagnosed by observation of behavior and cognition, as well as medical history. And it overlaps with other disorders. So again, autism, about 10 to 15% have epilepsy, about a third have intellectual disability. What makes autism distinct in its diagnosis is the problems with social cognition and what I call in a shorthand kind of mental flexibility. And again, shown below on the teeter-totter, you can have environmental factors or large effect size genes shown large or small effect size additive that all push the teeter-totter and push you over to the right in the red zone on these contours of language, social behavior, and repetitive restrictive behavior, which I call an alteration in mental flexibility, a resistance to changes in routines and et cetera. And one of the points I want to make is that the genes that cause autism don't necessarily, they're not like autism genes. They're genes that disrupt the brain function and development, leading to abnormal circuit development of the brain. So it's not surprising that something that disrupts brain development might not be that specific. In other words, you might have one patient with intellectual disability, another with autism and intellectual disability. They may even have a sibling with very, very mild epilepsy and intellectual disability, et cetera. But using the exome sequencing and a bunch of other technologies, including microarrays, we've been able to identify many, many genes. And so this is just showing what's happened over time. And you can see the explosion that's occurred that is coincident with that break that I showed you in sequencing. This new technology, next generation sequencing, now allows us to very inexpensively do an exome sequence on a patient. So it's now clinically indicated to do this. And there's just been a rapid bunch of technological advances shown below. And this was in 2008. That really, when things really began to take off. So now there are, in Safari, which is the Simons Foundation website, where they collate likely autism risk genes with different levels of evidence. There are now several hundred rather than just one or two in the database. So what this means from a genetic standpoint is there are many forms of genetic variation and modes of inheritance. Because we've identified hundreds, most of what's been identified are what are called de novo, like Down syndrome. Parents don't have it. It occurs in the sperm or in the egg, and it's called a de novo, not inherited mutation. It's still genetic. And if you find that, the recurrence risk to the next child is much different than if it were a dominant mutation or a recessive mutation like shown here. So there's autosomal dominant, right? We have these different modes of transmission, X-linked, like Fragile X or Duchenne's. And then there's the additive common risk, which still probably accounts for about 50% at least. Genetic testing can currently identify approximately 20% of mutations contributing to autism in most cohorts. So they're mostly de novo, but sometimes recessive or some of these others. And so genetic testing is indicated. We do a clinical microarray. It's a boy Fragile X, and we do exome sequencing. So what's happened is we've gone from 2008, where there are a handful of genes and not much interest in developing drugs, to 2016 and now 2020, where there are more than 200 candidate genes, clear mechanistic models, and their major drug development efforts in autism are now ongoing. And I think this is also happening as genes are identified in other neuropsychiatric syndromes. I'm going to skip, and this is just again from 2016, and what it's showing is that these rare mutations that occur, and the genes are shown to the left, some of them are single gene mutations, some of them involve regions of a chromosome. Those are called copy number variants when they involve more than 1,000 base pairs, and they usually involve multiple genes, sometimes just one gene, like Neurexin 1 deletion. And it's interesting because, Neurexin 1 deletion causes, is seen in autism, it's definitely increased, also in schizophrenia. So again, this overlap issue. But these disorders, all of them are rare, but when you add them up, they combine to about 10% of cases now, sometimes in some cohorts, almost 20%. But most of the genetic contribution is still predicted to be common inherited, and there's some that's unaccounted for, and we can discuss that later. But what we've learned from this is that there are going to be many genes. We predict maybe over 1,000. Highly additive effects in many cases, like I mentioned. None account for more than 1% of cases. And there's this thing called pleiotropy, which means that there's not a one-to-one relationship between gene and syndrome. Some of these genes, like CHD8 mutations, or better yet, that's SYNGAP1, that can cause just straight ahead intellectual disability, but it can also cause intellectual disability with strong autism, etc. And again, that's shown here. I'm just going to skip over this slide, because I want to make some other points. But I think I've made that point before, that there's not a one-to-one correspondence between genes. But in generally, those with large effect de novo mutations have more severe clinical courses, and that's shown here. With the more, if you have delayed walking, seizures, intellectual disability, if you have all three, the rate, the odds of you having one of these mutations goes up. So again, if you're in your clinic and somebody has language delay or seizures or delayed motor function or intellectual disability, the likelihood they're going to have a rare mutation in autism goes up past the 10%. It might even be 30%. Another point we need to say is that racial and ethnic diversity are critical. And right now, most of what we know is from European populations, a little bit from Asian. We have a long way to go. Here's an early paper from Zach Cohen's lab at Harvard, where they showed that if you don't have a balanced ethnic groups, you might call a mutation that is present, let's say, in African Americans who are typical without a disease-causing factor, not because it's causing the disease, but because of some ethnic ancestry issues. And here they show that several cardiac mutations that were thought to be pathogenic actually aren't. It just has to do with the proportion of somebody's ancestry from Africa. And so one of the things that we've been doing for the last 10 years now, we recognized this about a decade ago, is focusing most of our autism gene discovery work with our colleagues at WashU and Emory and Albert Einstein. We have an autism genetics and human diversity project where we're recruiting only African American families to add that group to the mostly European group so that we understand autism risk across different populations and avoid genetic misdiagnoses. So that's just one plug for that. But the point is, we've been, as a field, very successful in identifying genes. There are now hundreds identified in autism. And the issue is, how do we connect that to function? How do we develop therapeutics? And how do we understand mechanisms? And so one key point is that genes act through many levels of function to impact behavior. So the reason we study genes is because they give you a causal anchor. If you've met, the disease doesn't cause a genetic mutation, the mutations are where cause begins. And so, but DNA acts by, you make RNA from DNA, and then proteins are made within cells. The neuron cells form connections or synapses with each other that create circuits that are involved in behavior and ultimately the brain. And our job is to connect genetic variation, and it's through these various stages to its effects on behavior, which is daunting, but doable, we believe. And part of what allows us to do that is the fact that genes don't act in isolation, nor do cells, nor do circuits. They're part of networks, and we can identify networks by looking how genes act together. Genes that play together likely have the same function, and this is being shown below, where we take genes that are turned on and off together, and they get grouped into modules, and these modules correspond to the various cell types and from the ground up, cell types in basic biology. So we're able to take this very complex, tens of thousands of genes and basically understand how they work together in a few dozen pathways or modules. And this greatly simplifies the process to some degree and allows us to help begin to connect these different levels. So one of the questions is, if you have 200 or 300 autism genes that you've identified and you're predicting over 1,000 with large effect size, and then maybe hundreds or thousands with small effect adding up in some patients, these different models of autism genetics, how do you get your head around that? And so one question is, if you think about cancer, for example, are there defining principles? With cancer, all cancers involve proliferation, abnormal growth, and invasion. So do autism genes converge on some process in the brain? Do they act during the same time or in a particular cell type or circuit to impact behavior? And so now, having hundreds of such genes, we can ask that question, and we did. Where do autism genes act during development? And we do this by, first, we do genetic analysis. So we've done all the sequencing in thousands of patients. We've identified large effect, the larger the error, the bigger effect, de novo mutations that we presume are causal. We identify smaller effect, inherited mutations, and we ask, do they, across the genome, do they act in any particular biological process or time point? And we do this through network analysis. So what I've shown below is, let's say, a cartoon of all the genes in the genome. These aren't genes causing autism. We take all genes expressed in the brain during brain development, and we ask, how do they group together? How do they cluster, forming these modules? And then we can ask, are autism genes spread evenly across these modules, or are they enriched in a particular time point or biological process? And what we actually find is that autism genes indeed converge during early brain development. Actually, they converge right around between 12 and 24 weeks gestation, which is when the cerebral cortex is being born and connections are beginning to form. Some of these genes are expressed very early and involved in the process of building the cortex and then go down, and some come up and their peak might even be during the early postnatal period. And what we found in doing this, to summarize a huge amount of work and multiple papers, is that these genes coalesce during early cortical development. That means mostly prenatal, at mid-gestation, when neurons are being born. And they involve two major biological processes. The red genes are those that have large effects. Those affect global regulators that regulate the expression of other genes, called transcriptional regulation. And then the blue are those smaller effect genes for the most part, and those affect slightly later synaptic development, but they cross at around 16 weeks, and birth would be around here. So all of these genes kind of converge during early brain development. We can also ask, what kinds of cells are they expressed in? And we find, when we look at layers of the cortex, the cerebral cortex is layered, and these different layers have different connectivity. The superficial layers, layers 2 to 4, are the layers that connect your two hemispheres to each other and other cortical regions to each other. And these are where these genes are mostly enriched. So one could see, from a genetic standpoint, autism risk affects connectivity, which fits with a decade of neuroimaging research, which shows that long-range connectivity is disrupted as well. Most of that is for the large effect variation. Now, most genetic risk, as I mentioned, resides about half with common variation. Only about 20% have these rare de novo variants. So how do we find them? So the way the study is basically done is we get huge groups, usually thousands, this is just shown with heart disease, cases and controls, and we ask if we have a particular place in the DNA, and we do this. This is just shown for one base pair out of 3 billion, but we do this across the whole genome pretty much. And we ask, where are there regions of the genome or specific parts of the genetic alphabet or genetic code that are increased in cases versus controls. And so here's cases show increased C, right? And so that would be a genetic association. And again, as I mentioned, most genetic association, and this is just showing what's called a genome-wide association study, where this has been done throughout the entire genome, showing all the chromosomes from 1 to 22 here. And this is the higher the peak, the more significant. It's called the Manhattan plot, for those of you in Manhattan, because of the skyscrapers are positive. And so one finds these regions that are in red here, where there's a strong statistical evidence for association with the disease. Now, most of these regions are outside of genes. They're in this non-coding regulatory regions. So figuring out what these mean can be very difficult. Before, I was telling you about the de novo mutations. Those are identified because they disrupt protein specifically, that 4% of the genome. That makes it easy to interpret. But 95% of genetic variation is outside of that. So this was a seminal paper in 2014 from the Psychiatric Genetics Consortium, where they identified 108 schizophrenia loci. But most of these were in non-coding genomic regions whose function is not understood. They act not necessarily on the closest gene, they can act to very long range. So which of these are functional and what their targets were is totally unclear. In fact, only 10 instances out of the 108 were these actually in those protein coding exonic parts of the genome and could be interpreted. So what do non-coding regions do? And this is more for the scientists in the audience, but they turn genes on and off, and they splice them. They kind of make the different copies. And again, this is showing that these non-coding regions shown in yellow can be functionally annotated. We can measure what they're doing, and we can connect them to their genes of function using new methods. So if we can see if they're open and active, we can see what genes they're actually regulating. And this, what's called gene regulation, occurs in a cell type and tissue specific manner. So what happened, what affects liver may not affect brain and vice versa. About 40% of these things are shared, and 50 to 60% are tissue and cell specific. So this really creates a problem in that, especially for brain, we had what I'll say is no maps for these territories. We didn't know what these regulatory regions did. And this is taken, the title is taken from a documentary by on William Gibson, the science fiction writer, visionary. And I just love the title. And my wife and I were minimally involved in this movie, so I always think of this when I'm thinking about map making. So we recognized about 7 or 8 years ago, as technology became available to make these maps, that they were absent from brain, and especially developing brain. And because I just showed you that autism risk genes are enriched in processes occurring during early brain development, we needed to have maps for early fetal brain development. And so that's what my lab spent a lot of time doing. And so we ask a number of questions. We take fetal brain, we ask what genes are expressed there, what regions of the genome are active, that is controlling, turning things on and off. But it's not enough just to know what's active. You want to know what gene it's active on. And so I'll discuss that in a second. And then you want to know how they regulate them. In other words, you have a region, it's active, you find the target gene, and then you want to know what it does. And so what we found as we started to make these maps is that they can help identify through which genetic variants and the genes of their cognate genes. So gene regulation occurs through chromosome folding that brings regulatory regions in contact with their target gene in a tissue-specific manner. So here's showing a regulatory region on the left, that's that little bump. Gene A and gene B, this is not regulating, 50% of the time it doesn't regulate it, the closest gene. It regulates something farther away. It can be hundreds of thousands of base pairs away and many genes away. But what you can do is identify this folding pattern. You find these loops that occur. The genome is not just a linear array. It's a carefully packaged and folded three-dimensional. We can identify these loops and these regulatory relationships. And what we find is that only half of those loci in schizophrenia were acting on the closest gene. Many were acting on a gene you would never predict, far, far away. So we're able to kind of use these maps and show how important they were. We've now extended these maps, and I'm just referring to the publication below, during development, using multiple technologies, so that now we don't just have the chromatin folding, we can actually see what's active and open, and we can combine those together. And this allowed us to make the highest resolution map during brain development. And we use these to study brain evolution and disease. And I want to just show you a couple things, and then I'm going to end. So we now know what's active, we know what it functions, we know what the target gene is. To a first approximation, it's just, let's call it version 1.1, but it's better than having 0. We can now ask, we can take these genome-wide association studies and ask, where do all of the hundreds or thousands of risk variants for each of these genes lie? Are they affecting certain pathways? Are they in regions that are regulating fetal brain development or adult brain development, etc.? And so when we actually do this, we find that genetic variation within peaks most active in fetal brain and show risk for adult disease and brain size, and I'll take you through this. So what I'm showing here are a bunch of disorders on the left, from intracranial volume, educational attainment, not disorders, phenotypes, to some disorders like schizophrenia. And you can see that these studies range in size, and the larger the size, the more powerful it is. And so some of them, like the autism, are still relatively underpowered. But the schizophrenia is very well powered. And what we do is we basically map all the genetic risk onto things that are either active in fetal brain development or in adult. And we see that even for something like depressive symptoms, the PGC GWAS study, that when it's over the red line, it's significant. That is enriched in fetal brain peaks, but not adult here. Schizophrenia is about equally shared between them. ADHD, much more fetal than adult. Educational attainment, more fetal than adult, etc. So what I'm telling you is that, of course, some of this genetic variation acts, and when I say adult, it's kind of, it's hard to, we don't really distinguish between adult and childhood. So many of these would be active in childhood too. When I'm saying fetal, I'm saying prenatal. So it's really prenatal versus a couple years postnatal. It's really, we haven't filled in that middle gap, and that's going to be actually very, very important. So, but the point being that regardless of the adult vulnerability, there's substantial activity of these genetic risk factors during fetal brain development. I'm just going to skip this slide but this basically shows that it's in the neurogenesis. It's where neurons are being born that this really is acting. So another thing that we can do is ask, how does genetic variation actually act on gene expression? Like the genetic variation, I told you it's regulatory, turns genes on and off. And when we can do that by measuring the mRNA in the brain, we can measure the genetics across a lot of individuals and ask, how do genetic polymorphisms, these SNPs, let's say 1 and 2, affect gene expression? What is the actual effect? So here are 2 different SNPs, different genotypes, and this is showing that having a specific genotype, but these 2 SNPs has a substantial, increases expression, and the purple SNP, decreases it. And so these are called expression quantitative trait loci. It's basically how a particular genetic region has a specific quantitative effect on how much of that gene is actually turned on or off and made into RNA. So again, we did this, there was really no large-scale fetal brain map. And so a former graduate student in my lab, Rebecca Walker, did this with postmortem fetal brain, 201 mid-gestation, so at about between 16 and 22 weeks. And she ran this very complex set of analyses shown on the left. But I'm just going to make a few points here. One is that if we look, we can see that some of these overlap with adult, about 30, 40%, some amount, but that there are a lot that are fetal specific and some that are adult specific. Again, this is consistent with the need to have maps that correspond to different phases and stages of development and cell types. Another thing Rebecca's able to do is ask, okay, if I have an autism genome-wide association study, is it enriched in a particular set of genes or module of genes that's expressed? What I'm showing here is if things is expected versus observed, and there are about 10 different co-expression modules identified in the fetal brain, and one can see that most of them are along this line. Where what's observed is what's expected by chance. Except for this yellow one, which is way, way more, we observed way more enrichment. And that turns out to be these fetal upper neurons, just like I showed you before. Although here I'm using common genetic, it's a GWAS, genome-wide association, and before I was showing the large effect size mutation. So both large effect rare mutations, and common genetic variation act during early fetal brain development on development primarily of fetal, not exclusively, upper layer neurons. Another thing we're able to show, and again, this is for maybe aficionados, is that it's not just, this is EQTL, that's expression, this is splicing, how genes are sewn together, what form of the gene is made. And one can see that that also has an important effect. It's almost the same size. If we just look at the relationship of these disorders, one can see that the splicing QTLs also contribute to disease risk. In fact, EQTL, not significant for autism, but splicing is. So again, for these developmental disorders, ADHD and autism, it's not just how much of a gene is being expressed, it's how that gene is spliced together that's very, very important. And again, the fetal period is a big period of risk. We can ask the same question around schizophrenia, like as you see here. Schizophrenia has huge enrichment. It's the most enriched almost for both the EQTL, that is how high or low a gene is, and the splicing. So we asked, if we took fetal versus an adult, where is schizophrenia enriched? And this is showing over multiple size windows to show that our analysis is not arbitrary. And the higher the enrichment, the more significant it is. We can see adult shown here in blue. So there is some substantial enrichment in adult down here, but the fetal more so. So again, substantial fetal risk. And lastly, my last slide I want to show you. Let's say instead of treating these disorders, remember I told you in one of the first slides, there's a lot of overlap among the genetic risk for these disorders. And we know from looking at families that some families that these don't necessarily breed true, that you might have mom with bipolar, child with autism, etc., somebody else with anxiety. So now what we did with the Danish group in iPsych, because it's a population level, what they did is they got everybody with a psychiatric diagnosis in their medical system and genotyped them over a specific time period. So it's about 90,000 people. Cases and controls. And what we did is we asked if you had schizophrenia, bipolar, autism, anorexia, affective disorder, major depression, ADHD. If you had any of those, you were called affected. And that's the XDX, cross diagnosis. And what you can see is we were able to identify risk variants, shown here, these are 4 of them, that have pretty evenly distributed risk across disorders. Not exactly the same for all of them, but that affect 4, 5, 6 or more disorders. So in other words, there's risk that gives vulnerability non-specifically to a variety of disorders. And when we look to see where these genes were expressed, I'm showing the expression of the gene, the average gene. Its peak is at mid-fetal development in the cerebral cortex. And there was a second, this was in our first study, a second study done in a much larger group by the Psychiatric Genetics Consortium that we were also part of, also shows, again, pleiotropic greater than 2, those things that affect more than 2 diseases, again, peaking in the second trimester in mid-gestation. So again, there's strong evidence that early brain development is a critical window that creates vulnerability. And we have to understand why that is and what it's doing and whether or not and how we can modulate that later in life. So in conclusion, fine-grained understanding of gene regulation during human brain development is essential to understanding the functional impact of human genetic variation in the nervous system. That these approaches that I've told you about provide proof of principles and frameworks for understanding how genes are acting in the brain, defining points of biological convergence and disease, both in terms of pathways and cell types and circuits. Autism risk genes converge on early fetal brain development in the peak of neurogenesis, involving glutamatergic neurons primarily. Some of them are broad, but if you want to know where they converge, it's on these upper layer neurons. And I think understanding which genes and cell types are impacted provides a new starting point for developing therapeutics. So thank you for your attention. I'll take some questions now if there are any.
Yes. First of all, thank you for an outstanding presentation and the work that you do. These are complicated issues, and I want to thank you for explaining them in a user-friendly way. One question that has been asked in a variety of ways is, where do you see this research going in terms of potential treatments for people and potential methods of prevention? What do you see happening in the next period of time?
Yeah, so I'm going to divide that into two basic things, which is, one is the question, if these things are acting in early development, can we do anything after somebody's born? I'm an optimist, and there's evidence from animal studies that the answer to that is yes. We first have to understand what circuits they're impacting, what cell types, and then I think we'll have ways of modulating those cell types and circuits quite specifically down the road. So I'm hopeful that we can do that, but we have to understand what the mechanism is. We can't just put our head in the sand. So that's number one. Number two is that with new viral technologies and CRISPR and other forms of editing, and there actually are going to be now we're using nucleic acid therapies, either RNA or DNA and virus, to treat a number of diseases, from sickle cell to spinal muscular atrophy, Duchenne muscular dystrophy, et cetera. And these are actually in the clinic now, and they're going to explode because they're effective. We have ways using CRISPR of turning genes on using their native promoters. With most of the autism risk genes, even those having large effect sizes very early, most of them, not all, actually only affect one copy. So they knock out one copy, and that's enough to cause disease. Well, how about if we turn on the other copy doubly? And so we have ways of doing that now. And actually in my lab and in others, we have ways using CRISPR of activating those loci. And so we've been able to show in unpublished work activation of multiple haploinsufficient genes. So one could turn these genes back on. The critical thing will be knowing when and where to do that. But I think we'll be able to do that. And I think that's it kind of sounds a little science fiction-y, but I really, that's where we're going with regard to these therapies. Thirdly, and I didn't discuss this, it wasn't part of this particular lecture, but I could have driven it that way. We work a lot using stem cell models now, these 3D organoids, with a variety of collaborators and in our own lab. And there's a whole, the field has really adopted these, and they show great promise. We can put mutations in them, and we can understand the mechanism using these in vitro models, like these little mini brains in a dish. And we just had a paper with Sergio Pasca at Stanford on the 22q deletion, which is a very, very complex deletion, showing that we can actually identify the mechanism, and by identifying the physiologic mechanism, we can reverse it in that system. That provides a starting point for therapeutic development. Of course, that's all preclinical. There's a lot of work that have to be done to translate that into a drug. But again, I'm very hopeful that the kind of three approaches I just mentioned to you will be implemented or are implementable.
Well, that's extremely exciting and offers tremendous hope. And I think is an example of how very basic science can ultimately be translated into clinical care and clinical interventions. So we're going to look forward to hearing about that, hopefully in the not too distant future. I want to again thank you, Dr. Geschwind, for your presentation and for your work. And I want to thank everybody for joining us today. 100% of all donor contributions for research are invested in our grants to scientists. All of the research we fund is made possible through supporters like you. So please consider making a gift by visiting our website, bbrfoundation.org, or calling us at 1-800-829-8289. This webinar has been recorded. If you've missed any portion of the presentation or would like to share it with family and friends, please visit the Events and Webinars page on our website. I hope you'll join us again next month when Dr. Stephen Hinshaw, Professor of Psychology at the University of California, Berkeley, and Professor of Psychiatry and Vice Chair for Child and Adolescent Psychology at the University of California, San Francisco, will present ADHD, Controversy, Key Research Findings, Rising Prevalence, and Promise. This webinar will take place on Tuesday, January 12th at 2 p.m. Eastern Time. I want to again thank everybody for joining us and wish you and your family a happy, a healthy, and a safe holiday season and new year. Once again, thank you.